talk about some history of microbiology. So what I'm going to be testing on is who did what in microbiology early on. I will ask you something about these guys and let you know kind of what they did. Now, one of the things that I will be doing is not dates. Now, the dates might be part of the description, but I'm going to ask you some things. So we're going to start out with the history, and basically the bacteria, the ancestor of bacteria, that probably was the first life on Earth, evolutionarily speaking. In about 1665, Robert Hooke reported that living things are made of cells. He viewed a piece of cork in a microscope and said, these things are made of cells. He saw the first cells. They were dead cells at the time, but he saw them. Now, the first microbes were actually observed in about 1673. Then in 1858, Rudolf Virchow helped get the first part of cell theory, that cells arise from other cells, pre-existing cells, which helped to lead to cell theory, that all things are made of cells and they come from pre-existing cells, part of the modern cell theory that I've given before. Now, a guy named Anton van Leeuwenhoek. Leeuwenhoek was this really fascinating guy who this Dutch guy probably was autistic. He made over 400 microscopes. He's called the father of microbiology. One of the first to ever use a microscope. He was a Dutch merchant but amateur scientist. He made over 400 microscopes. The first to actually see live microbes. He would get rainwater, feces. He would collect his diarrhea. He would scrape his teeth and the teeth of people who didn't brush their teeth well. He would do all this stuff. He sent his stuff off to the Royal Society of London, which at that time is kind of like the National Science Foundation, but for London, and they still exist today. The Royal Society is a big deal. And Robert Hooke confirmed his findings. Turned out they were trying to figure out how come his microscopes work so well. Found out after doing some work that actually some of his stuff was based on Robert Hooke's work. Now, let's talk about this bit of history, is this idea of how diseases came about. There's many ways that we thought things came about. Now, we have the germ theory today. Germ theory of disease. Now, there were loads of ways on how things came about. Spontaneous generation was one of the ways people thought that life came about. That life came from non-living matter because a vital force was present. A vital force formed life. Maybe that was a god or something. Now, with this came this idea during the Black Death, cholera, chlamydia, and the Black Death. If you've ever seen the famous masks the plague doctors wore, that's how I dress on Halloween. My Halloween costume usually is a plague doctor. I'm very fascinated by the Black Death. It was because miasma theory. Miasma is this idea of bad air, a noxious air, bad air. So that beak, they put good smelling herbs, ring around the rosy. That was the sores that would grow on you. Pocket full of posies that prevent the bad air. Ashes, ashes. We burn the bodies. We all fall down. Everybody died. So many people died from it. Ring around the rosy, pocket full of posies, ashes, ashes. We all fall down. The children's nursery rhyme is about that. Bubo comes from bubonic plague, sore, the buboes. A boo-boo. You got a boo-boo. You have the bubonic plague. Good for you. All right. Now, miasma theory was that prevailing dominant thing. Now, no one would have had, with any sense, if I went to them and said, no, really, most disease is caused by these little things you can't see. That's what makes you sick. They'd be like, you're crazy. They wouldn't believe me. There was no evidence. The evidence for miasma was stronger. So what happened was, guys, it took convincing. To get us there from this idea of spontaneous generation and miasma, we go to biogenesis, the hypothesis that living things make life. Life comes from life. Rudolf Virchow did this. But let's look at some things that gave us in 1858, Rudolf Virchow really put it all together. But let's look at how we got there. Back in 1668, this guy named Francisco Redi, he took 6 jars and put decaying meat in it. 3 jars were covered with a fine net and there were no maggots. 3 open jars and maggots appeared. Where did those maggots come from? Well, the flies landed on the meat that was open and laid the maggots. And the sealed jars that had a fine net kept the flies from getting in there, so no maggots showed up. The maggots didn't show up from a vital force. If that meat had vital force, the meat in the covered jar and the meat in the open jars, they have the same vital force. Maggots only came when flies could land on the meat. So that would have been biogenesis, okay? The life coming from flies. Flies laying the maggots, okay? Flies laid their eggs on the meat. The maggots showed up later, okay? John Needham in 1745 took a broth and boiled it, which would have sterilized it, put it in covered flask. Nutrient broth was heated and then placed in a sealed flask, and there was microbial growth. The microbes were sealed what he thought was a sealed flask, but it got in, okay? From when he poured it in. He heated it and then he placed it in the sealed flask. When he poured it, it got in. Spontaneous generation? No. Biogenesis. It was contaminants from the air and he transferred it. Lorenzo Spallanzani, he boiled a nutrient solution in a flask. The nutrient were placed in a flask, heated and then sealed. There was no microbial, so it was placed in a flask. Then it was heated and sealed. It stayed clean. It never got exposed. It didn't get any kind of infection. Now, Pasteur came along and really did a great experiment. What he did is he had 2 flasks. He had one flask that was heated and not sealed, and another one that was heated and sealed. The one that was heated and not sealed had this S-shaped neck to it. And what would happen is it would let air in that had bacteria, and microbial growth happened. But when he sealed it, it was boiled and sealed, no microbial growth because it actually was sterilized. So this is biogenesis. Again, favor of biogenesis. And the theory of biogenesis, Pasteur's S-shaped flask, really showed that, hey, this broth, which was sterilized, both were sterile, but one that was open to the air, bacteria could get in. Okay? All right. So that's how we got to biogenesis. Okay? It was all these guys' work. So know these guys, what they did. Okay? With their experiments and how that got us there. Okay? Now we're going to get to the golden age of microbiology. This deals with a lot of people like Louis Pasteur and things like that. Okay? Now, this is right around 1857 to 1914. It's called the Golden Age. Pasteur's work, microbes and disease and immunity and antimicrobial drugs, all were coming out at this time. So this was really big. Okay, Pasteur showed that microbes did fermentation. Fermentation is what makes beer. It's sugar to alcohol, beer and wine. It was showing that we would get wine get spoiled and make it into vinegar, and it would spoil the wine because bacteria got in and food spoilage. This was able to make these foods last longer and the wines last longer. We were basically seeing there were microbes and diseases. We were getting immunology understood, and the first antimicrobial drugs were getting made. Fermentation, we turn sugar into alcohol. It's done by yeast, and they do this when there's no air. We use it to make beer and wine. Pasteur was the one that proved microbes were doing that. He also showed that microbial growth is what caused food to go bad. Alcohol gets converted into acetic acid. The bacteria in the air get in. It makes vinegar. It would ruin the wine. Fine wine would get ruined. Okay? Pasteur said that food spoilage was done by bacteria, and if we could heat it, not enough to evaporate the alcohol, but enough to get the wine, so the wine stayed clean. Okay. Pasteurization was the process he made. It was a low heat, enough to kill off the bacteria, but not destroy the thing. Now, does pasteurization, sometimes people say that it takes away some flavors, but it also reduces your risk of infectious agents, of communicable diseases. Okay? All right, germ theory disease. How do we get to germ theory? From miasma to germ theory. Well, one guy, 1835, we had this guy named Agostino Bassi, or Bassi. Bassi showed us that a silkworm disease was caused by a fungal infection. Louis Pasteur said there was another silkworm disease caused by protozoan. Why do people care about silkworms? Silk was a big thing that we traded in at that time. Silk was a big thing. Made ties, silk handkerchiefs, part of fashion. Silk was a big part of the fashion of the times. So big deal with silk. So it was a big thing. Comes from silk was made by a silkworm. They would dye it, turn it into all kinds of silk products. Silk was heavily used. Okay, Ignaz Semmelweis was hand washing prior to like childbirth obstetrics. Like they would go one patient to another and transmit it, or they would go from dissecting cadavers. But they wouldn't wash their hands from patient to patient. There's another thing that happened, okay, called puerperal fever. Puerperal fever, okay. Pasteur's work showing that microbes in the air can spoil food and cause disease. A guy named Joseph Lister came up with a disinfectant. You may have heard of Listerine, okay. Is another one of these things named after Joseph Lister, okay, to prevent surgical infections. He did that. Now, a big guy in 1876, Robert Koch, he proved that bacterium anthrax was causing a disease. That the disease of anthrax, anthrax is caused by a bacterium. He came up with what's called Koch's postulates to show that this is the microbe that causes this disease. And he set upon his set of postulates to do this. We will learn these postulates later. Okay. Now, vaccination. A big one is Edward Jenner. Know that name. You'll definitely see Edward Jenner at some point. This first was against smallpox by giving a person, inoculating with cowpox, protecting them from smallpox. Vaccination gets its name from cowpox. Vaca means cow. Okay. Protection is called immunity. Immunology is the study of that. A vaccine is we might get an avirulent organism and inoculate them against a really virulent organism. We may also use interferon, chemicals that we make to inhibit viruses. This is all examples of chemotherapy. Chemotherapy isn't just used to treat cancer, it's used to treat microbes. Chemotherapy is a synthetic drug like an antibiotic as well. Antibiotics are made by microbes to fight microbes. It's made by bacterial and fungi to kill other microbes. It is microbe competitor stuff. Okay? It's like, I'm this bacteria. I want to be the bacteria that wins. So I will produce a chemical that makes the other bacteria around me lose. Now, let's talk about some chemotherapy agents. Quinine, tree bark, used to treat malaria. Paul Ehrlich was like, there's got to be a magic bullet that can kill the pathogen but not harm the host. Now, for a lot of pathogens, if we're dealing with something like a worm infection, the worms, they get in, they are eukaryotes. What will harm them will harm you. So dewormers are pretty harsh stuff to use sometimes. Ehrlich used a synthetic arsenic drug called salvarsan to treat syphilis. That's in 1910. 1930s, you had sulfonamides. Very early antibiotic. Sulfonamides. They are bacteriostatic. But here's the big one. Alexander Fleming in 1928, the first big antibiotic. Penicillin, the true magic bullet. This thing from a fungus called penicillium, it was a happy accident. It was a fortunate accident. The penicillin colony got in and was like, hey, this is killing this. It's like, wait a minute, there's something going on here. There's an area of inhibition. It was a happy accident, a fortunate accident, an accident that led us to see that penicillin bacteria made an antibiotic that killed Staph aureus. It was tested clinically and mass-produced, and we were able to do that. Now, microbiology has given us a lot of things. One of the things microbiology has given us is bacteriology to study bacteria, mycology to study fungi, virology to study viruses, and parasitology to do protozoans and worms, parasitic worms. Okay, pretty easy, those terms. Okay, now, Rebecca Lansfield, a great woman of microbiology. Immunology, study of immunity. Vaccines and interferon, we are trying to look at them for viral diseases. Vaccines and interferon. Interferon is used to treat viral things. We can look at some bacteria has serotypes to them. And the use of immunology to study the serotypes, subtypes, things that we can use to say you have this substrain that we can treat differently. Rebecca Lansfield, this is the 30s, 1933, big, she's a big woman, big famous woman in microbiology, Rebecca Lansfield. All right, modern developments, we have microbial genetics. We study microbes and how they inherit their traits. It's really fascinating. Microbes can take the genetic material from the environment, bring it in and use it, and be like, oh, I'm now antibiotic resistant now. Molecular biology is the study of how DNA directs protein synthesis. Genomics, that is the genes, all the genes of a microorganism. We can do the whole, understand all their genes, what's going on, and we can use it tools to classify microorganisms, their genome. Recombinant DNA technology gave us the ability to edit DNA of bacteria, put stuff in bacteria to do things like make insulin. Bacteria produce animal proteins, like bacteria make insulin for us. That was like Paul Berg in the 60s did that. Okay. All right. Now, several people in 44, Oswald Avery, Colin McLeod, and Macklin McCarty, they all did the DNA was a hereditary material. Okay. In 53, James Watson and Francis Crick discovered the DNA structure. Paul Berg in the 60s did recombinant DNA technology, and he basically took animal DNA and inserted it into bacterial cell. I've done it. I've put animal, I used to put the green fluorescent protein from a jellyfish into my bacteria, and then with it I would put an antibiotic resistance gene with it, and so I would know my bacteria antibiotic resistant because they would glow in the dark. We also have microbial ecology, that study of relationship microbes in their environment. We're trying to understand that because there's loads of microbes that are out in the environment. That's where we're looking for the new antibiotics. We're finding new species by digging in the dirt, going out in the Appalachian Mountains, and trying to find microbes that are not only new, but making antibiotics. Okay? Bioremediation, we can break down sewage, oil, even mercury can be broken down, contaminants, by getting rid of pollutants. Biotechnology can make microbes produce food and chemicals like biodiesel. There's a lot of emerging microbes. Microbes that are pathogenic to insects could be something that we use instead of pesticides. Can prevent insect damage to crops without having the stuff being in our food. Okay. Bacillus thuringiensis infects insects but harmless to animals and other plants. Okay. Now, when it comes to infectious diseases and their resistance, and it's emerging, is resistance when the pathogen overcomes their host resistance. Okay. I want to show you something here really quick in a minute. I'm going to pause and show you something in a minute. Resistance when a pathogen comes into host resistance and the disease results. If it overcomes their host resistance, the host stuff that they use to fight the disease, their immunity. So emergent infectious diseases, they're new, and we don't have antibodies against them. We won't have like we may not be genetically predisposed to fighting it. There are people born immune to HIV. New diseases, or if they're new or they're increasing in incidence, they're called emerging. Okay. These new emerging infectious diseases may be resistant. There may be resistance. Now I'm going to pause and show you something here. Okay, here we are. Okay, so I'm going to play this video here. I want you to watch this. Mega plate, a petri dish.
The building is basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, pour some thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads, until it starts to compete with other mutants around it.
And they're producing antibiotics themselves.
When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type.
11 days!
And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
All right. So emerging infectious diseases can be because they've been around and they've been normal, but penicillin or other antibiotics become resistant to it. MRSA, methicillin-resistant Staphylococcus aureus, highly resistant bacteria. By the 1950s, penicillin resistance had already developed. 1980s, methicillin. 1990s, MRSA was resistant to vancomycin. Then there's vancomycin intermediate resistant Staphylococcus aureus. Vancomycin resistant bursa. And we're going to get to the point, guys, where we're going to go back to roots and tubers to try to fight these things. We need to find new antibiotics. We need to find them now. Okay? All right. Now, some other emerging diseases. Now, this bovine spongiform encephalopathy, this is actually caused not by a virus, not by a bacteria, but by a misfolded protein called a prion. Mad cow disease we call bovine spongiform encephalopathy. In humans, it's Creutzfeldt-Jakob syndrome or disease. Now, beef infected with the prion, we get it. Now, there is a variant where you get it and you can transmit it by bites. Zombies. There's West Nile encephalitis. That's an infursing West Nile encephalitis. It's viral. It's transmitted by mosquitoes. Cryptosporidosis. It's the diarrhea caused by a Cryptosporidium protozoan. Contaminated water. 1993, Milwaukee had over 400,000 people sick. AIDS, HIV, that was terrifying when I was a kid. HIV, AIDS was terrifying. HIV, the human immunodeficiency virus, worldwide epidemic still. Over 6,000 new infections every day. But when I was a kid, it was a death sentence. Now, for anybody who knows me, I don't know if you can see it, but I have a scar on the back of my head. When I was in middle school in the 90s, I had fallen. So in elementary school, I had an accident on the playground and I broke my clavicle, broke my right clavicle. It would never heal, it would never heal, it would never heal, it would never heal. They could not figure out what was going on. I had all these problems and they couldn't figure out why I had all these problems. They sent me finally, years and years later, middle school age now. So this happened in first grade. I broke my clavicle in first grade. And then what happened was, in middle school, I went in for some screenings. And they sent me to the Shriners Hospital. The Shriners sent me to the hospital next door. And I spent all day in an MRI. Head to toe, they MRI'd me. They did every test they could possibly do on me. The head to toe MRI discovered that my cerebellum had herniated on my foramen magnum. And they were going to have to do surgery. And at that time, it was one of the things that happened to people is sometimes they get HIV from blood infusion. Because they did not want to give a kid HIV. And it happened. Ryan White got it in the 90s. What they did is they brought me in before the surgery. I gave blood to myself. Then they did the surgery after that. So I actually donated blood to my own self so that if I needed blood, I'd have it. Because they didn't want to risk giving me HIV. It was that big of a deal at that time. It was terrifying. And if you got it, it was like, it was terrifying. It was a death sentence. Okay, we're doing a lot. We made tremendous. It destroys your T cells. Your immune system is wiped out. You die of some random disease nobody ever gets anymore. So it is an STI, STD. STI, males and females both get HIV/AIDS. COVID-19, the disease caused by SARS-CoV-2, severe acute respiratory syndrome, coronavirus 2. Official name, SARS-CoV-2. That's the virus. Responsible for COVID-19, the disease. COVID-19, coronavirus disease, 2019. We all know, we probably remember the pandemic. Okay. Coronaviruses are common cause of colds and upper respiratory infections. This nasty little COVID-19 virus, what it happened was, it got something to its RNA that allowed it to replicate at warmer temperatures, because normally these coronaviruses have to go in your ears and cooler parts of your body, in your head to get a head cold, not lower respiratory. Okay? It was a really weird disease. Let's talk about normal flora. Normal flora, these are your bacteria. We used to call it flora because we actually once upon a time thought bacteria was related to plants. So they used flora for microbes at that time. In microbiota is what we oftentimes, we still call it normal flora or normal microbiota, your bacteria. You are more bacteria than you are you. There's more bacterial cells on you than there are your cells. You're more bacteria than you actually are you. Okay? Very important is this bacteria. And they colonize the host without causing disease. They actually can help. They can prevent growth of pathogens, like the bacteria that cover the gut prevent C. diff from going out. They can produce growth factors that help us heal. They can produce folic acid. They can produce vitamin K for clotting. They can help us in resistance. They can ward off other diseases like C. diff. Mucous membranes, stomach acid, antimicrobial things in your skin can help you resist bad diseases. These can all help you keep from getting sick and actually make you healthy. You need your normal flora. It's there. It's part of you. I want to talk about biofilms. In your teeth, you get a biofilm. This is your microbes that grow in rocks, pipes, teeth, and all that. Medical implants grow inside pipes, called a biofilm. Staphylococcus make biofilms, things like that. That's about mumps virus. This is a mumps virus here. Our mumps virus infects your parotid gland, saliva, and close contacts. You need 2 doses of the MMR to be 88% effective. The NHL got it in 2014. It broke out. It broke out at the University of Illinois. Is all the time getting it in camps, colleges, schools, get mumps outbreaks. Chikungunya. There we go. Chikungunya. I say it right. I practiced it this morning. Chikungunya. It's an African virus. Aedes aegypti, some of the Aedes species, Aedes aegypti. It's actually gone to Caribbean and Florida by now. Causes fever, joint pain, headache. Most people get over it in a week, but chikungunya. There we go. Now, measles, it's a viral thing. It's outbreaking right now, big time, due to lack of vaccination. Largest outbreak in 2 decades was in Ohio back some time ago. Unvaccinated travelers to the Philippines, they spread it. Outbreak in Disneyland in California. Don't confuse Disneyland with Disney World. There's been recent outbreaks in America due to lack of vaccination. Get vaccinated. Cough, fever, runny nose, rash. Starts at your hairline, moves down your body. There is a vaccine. Get it. I'm vaccinated. Do it. I need to go back and get my an updated vaccine. It's been long enough. Me being an elder millennial. Zika virus, Aedes aegypti. Aedes aegypti does a lot. Aedes aegypti transmits chikungunya, some other diseases. It can spread from mother to offspring and by sex. Zika, microcephaly, Guillain-Barré can also, which is a demyelination disease. If you remember that from my A&P1. Latin and South America, documented cases in Florida and Miami, Florida. All right, polio. Polio is a viral infection. Infects your brain and spinal cord. It can lead to paralysis. Syrian children got it due to the civil war in Syria. 91% vaccinated. Syria went from 91% vaccinated to 68% vaccinated due to the civil war. And this was a widespread worldwide. We tried to eradicate it. Still outbreaks in Pakistan, Afghanistan, and Nigeria. Polio. Poliomyelitis is nothing here. FDR had it. A president who got it was paralyzed by it, or at least wheelchair bound by it. All right, that contributes that, and we are done. And I will see you in the next one.